my father is blind. My father is actually blind. So when Brian, when I learned about what Brian Shaw is doing, he's going to tell you about it in a minute, I was really intrigued. So Brian has a son who also is visually impaired. And so Brian's desire is to help students with visual impairments or other disabilities be able to participate in science and do things that people used to think they couldn't do. It used to be that people with a disability were excluded from science and any whole number of things. Well, now doors are opening up. So someday if you have a child or a relative who has a disability, because of the work that people like Brian are doing, there's going to be more jobs that they can do and more activities that they can participate in. So Brian Shaw is here with 3 of his students in the back of the room. And Brian, I gave a very brief description of what you do, but we'll open it up to you. Maybe introduce your students who are here with us as well, and then we can.
There's 3 doctoral students in my lab, so they're getting their PhDs in biochemistry.
Sure. So my name is Levi. I'm a graduate student in Dr. Shaw's laboratory. And so growing up, I essentially wanted to be a dentist, but then I kind of got grossed out by the fact of looking in people's mouths. So I wanted to switch that and do more sciencey work. And so in Shaw's laboratory, we work with proteins. And as he was mentioning, proteins come in different sizes and shapes. And specifically, we work with proteins that have metals in them, like iron and copper. And so we work with these proteins that have these metals that have electrons that can move from protein to protein. And we study how different mutations of these proteins can affect the way in which they do their job. And so I work with a protein called RISC-E, and it's in each and every one of you right here, and almost on every animal on the planet. And so by studying this, we can see in which ways we can mutate it and amplify and attenuate the way in which it does its job.
Why does that matter? Why do you do that? What's the end result of you learning those things?
Well, we work with these proteins, not necessarily the ones that work with like niche things. As I was mentioning, this RISC-E protein is in all of us. And so by getting an understanding of how these different metalloproteins do their job, that essentially gets like a broad spectrum to all the different metalloproteins and how they do their job. Now this RISC-E protein helps us make energy. And so if it doesn't do its job properly, that can be very catastrophic. And so by seeing ways in which we can inhibit the way it does its job or amplify the way in which it does its job, that can kind of further our knowledge into a broader spectrum.
So I'm Emily. I've always kind of liked science, so I guess it kind of fits that I ended up going into studying science. How I ended up studying biochemistry though, I went to a small undergrad. The actual major I want, they did not have. And so I said, wow, biochemistry looks fine. And that's how I got into biochemistry. And then I ended up really liking it. And then I did like a summer undergraduate research experience because I went to a small undergrad, so we didn't have a lot of research there. And I worked with like some really cool microscopes that were huge and were like $500,000. And the pictures looked really pretty. And I was like, research is cool. And so that's when I started looking at research and applying to some different labs, well, schools. And I saw that Dr. Shaw, his lab works with the blind and visually impaired. And so I was like, that looks really cool. So that's how I got interested in the lab. I actually don't work with proteins. I work with DNA and RNA and looking at some different modifications to that and then figuring out how it affects transcription and translation. I know that there's some cell division models over there, so I figure you guys probably know a little bit about cell like division and multiplication and stuff, and how like DNA is like what makes you you, right?
I'm Travis. When I was at my undergrad at the University of Wisconsin-Madison, I actually was going to become an engineer because my older brother was also an engineer. I was going to get my degree in chemical engineering. I actually failed a statics class, which is like just a really tough physics class. The engineering school kicked me out and I was like, man, what am I gonna do in life? I looked at like the credits that I had and I was like, I'm close enough to get like a biology degree. And then I realized, wait, I'm like one class short from like biochemistry. So I ended up getting my degree in biochemistry. And then when COVID hit, I was like, man, what should I do? And this research company hired me and I was like, wow, I really like the lab. So then when I started looking around, I was like, what can I do with like my degree? I realized I wanted to get my PhD. And like Emily, I was kind of like looking around and then I saw Dr. Shaw and I saw his research with the blind and visually impaired and I was like, I want to do that. Like, it's so cool. I get to do 2 things. help the blind and visually impaired community, but also do, chemistry, biochemistry. And what I do in the lab is I kind of do a lot of things similar to Levi, but I work with two different proteins. So I work with azurin, so it's a blue copper protein, and then I also work with cytochrome C, and it's a red copper or red iron protein.
And we're going to tell you about proteins today. So, do you know what proteins are? Anybody know what a protein is? Anybody want to give us a quick little definition? Yeah?
That's right.
Sure.
It's kind of like things that build muscles, and
Yeah.
you take a bunch of them, builds muscles, and puts on mass.
Yep.
And
And then some proteins cause human disease. So let me walk through here and just see this one here. This nasty little protein is called alpha-synuclein, and it actually causes Parkinson's disease. Do you know anybody who has Parkinson's disease? They sometimes tremor and they get shakes. That's what you notice. You might have grandparents or even aunts and uncles who have it. So studying the structure of this protein, why it behaves the way that it does to cause this disease is something that we do in the lab. Now another one of these proteins, I believe this one is cytochrome C. I think it is. We'll leave that one. But one of you had, I think one of you had copper zinc superoxide dismutase. Let me see. This one here binds a copper and a zinc. It actually causes ALS. Has anybody ever heard of amyotrophic lateral sclerosis? ALS it's a pretty rough disease. Your muscles slowly waste away and you become paralyzed. Does anyone know who Lou Gehrig is? Babe Ruth's teammate on the New York Yankees. The disease is named after him. He was called the Iron Man in baseball. It was just awesome. Every season, he just nailed it. Super high batting average, tons of home runs, and then one day, just started going down. You can see his baseball stats literally just started plummeting almost overnight. Turns out he got ALS. So we study this protein, why it might cause ALS. We try to figure out what is it about this protein that causes motor neurons to die, and then we try to come up with ways to stop it. So proteins are really important. Models of proteins. And what you'll notice is a protein is actually a long string of atoms. It's a string. You can kind of see that here. And you see some of these little coils? They're called alpha helices. So a protein is a long string. And you're out what its structure is, right? So that we can come up with maybe little drugs that bind to it and shut it down. So if we ever want to study one protein, we got to separate it from all the other 100,000 proteins. And so those little boxes over there, they help you do that. But they're kind of hard to use. So we're actually going to see if you're able to do some of this stuff. Okay, we're going to have to take tiny amounts of liquid, microliters. Does anybody know what a microliter is? What's a microliter? 1 millionth of a liter. So you probably think about like 2 liters of soda. Hopefully you don't drink that much, but you at least see it at HEB. So 1 millionth of half of that, tiny amount. Okay, it's hard to see. It's hard to even handle that tiny amount. But we have special devices over there called micropipettes. They're these handheld things that sort of look like ice picks, and they're going to help you pull up tiny amounts of liquid that we have over there and load it onto this device that will help separate proteins. Okay, so if you're a biochemist, or if you're a biomedical researcher, or a pathologist, or a virologist, you could be doing this stuff all day.
You're gonna open up your Epinerf tube. It is just glycerol, so it's sugar and a colored dye. You're going to, so if you feel the pipette tip top, it has 2 stops. So 1, 2. You just want to go down to the first one, submerge your pipette in the solution, suck up, So now you guys can see it. And then when you're ejecting it, you want to go all the way down. So if I'm in the well, I would hold it in the well, and I would go 1, 2. Follow?
With visual impairments like my kid, we don't want to keep them out of the lab early. So Levi over here invented, I helped a little, but it was mostly Levi, invented this device. We call it a Zamponia. Zamponia, we named it after the South American pan flute. You ever seen it? This device will snap on to your gel box, and it will guide your pipette to a perfect position in each well. So whether you have vision or not, it helps you. We made it asymmetrical. We made it go down so you can feel, you always know which way's left and which way's right. So if you snap this on, the students will help you, the graduate students will help you, onto your device, it's gonna make loading it so easy. So let's give that a shot. See that? Well, if you're a person with blindness, how are you going to be able to analyze that data? Turns out we use an ancient Chinese art form called the lithophane. See how it's like an engraving? But when you hold it up to the light, it's a picture. See that? You ever heard of lithophanes? This is Aretha Franklin, the famous blues singer. So we have lithophanes of the gels. Here. Everything you can feel, you can also see. So it's what we call universal design. Anybody, regardless of their level of vision, hold that one up to the light. Anyone can visualize it. When I was your age, I wasn't really good at school. I was a wrestler, and I played football, and I pretty much just liked sports. But I did get into chemistry when I was a sophomore in high school. And when I took my first chemistry class, and I realized that chemistry really explains how everything works, I got really interested. And so it was chemistry that did it for me. But I think I graduated with like a 2.9 GPA. I wasn't a very good student. Went to a state college. I think I barely got in. But there I got into the lab, and that's where things I just got really into science. Then I ended up at UCLA, and then I finished at Harvard for my postdoc. So started out pretty low, but because I got interested and I worked hard, just kind of slowly went to the top. So matter. Science is for everybody. You're born scientists. You are born scientists. You're always asking questions. You want to know why stuff works. And sadly, the world sometimes works on you to stop being scientists. So don't stop. And the universe is an amazing place, and we know very little about it. And there's just so many jobs and opportunities for you. If you ever want to go to graduate school, that's college after college, and get a PhD, it's paid for. You don't have to pay any money, and we actually pay the students. So you get paid to do it. And it's great. You can work on whatever part of the universe you want to study.